Next to Plant-DiTech from Israel and Catherine Jacob.
Thank you, Francois, and welcome to Plant-DiTech's presentation. My name is Katrine, and I'm actually in the San Francisco Bay Area presenting Plant-DiTech here in the United States. Our technology is a plant phenotyping technology, which measures a plant's reaction in its different environment. Our technology is suited for plant breeders, for product developers, who can test their formulations, their product candidates, as well as their genotypes and breeding populations, particularly under abiotic and biotic stresses, fast, accurately, and reliably. Our platform has three main features. It can precisely control your experimental conditions. We measure directly plant physiological yield-related parameters, and the data are instantly available by cloud-enabled and statistically analyzed. So you can really shorten your testing cycles, both for your products and for your breeding genotypes and populations, because you see the data instantly, you know what your plant is doing right away in your experiment. The technology has been developed out of the Hebrew University of Jerusalem by a real problem. The two professors, a plant physiologist and a soil scientist, came together because the plant physiologist could not see a phenotype in aquaporine mutants. And generally in plant phenotyping, there are many, many other challenges that are still out there, particularly if you want to test under stress conditions. It's very hard to precisely set your conditions so that you can actually repeat your experiments, that they are reliable and comparable. It's very hard to capture the real genotype by environment interaction because conditions around the plant change constantly and the plant is reacting also instantly to different conditions out there. Yeah. So our Plantarray platform has set out to solve some of those challenges. As I mentioned earlier, part one is we can highly customize water and fertigation to each single pot. On the right side, you can see that we are able to capture the plant environment interaction. We measure the environment, we measure the soil, we measure the plant, and you can see we are able to directly measure biomass. We measure plant transpiration, stomatal conductance, water use efficiency, and we also measure root activity, among other parameters. How does the technology work? The backbone of the technology is that we can precisely measure the water flow from the roots out through the shoots. And you see the patented technology is a set of plastics which enable that we can precisely measure, and also setting up the conditions for your plant so they can grow in those conditions that you're setting it up for. Each unit has its own controller, and that allows at one side for the precise delivery of your conditions and also for the precise measurement. We measure constantly and we measure simultaneously. We measure every 3 minutes and we measure at the same time for each plant in the array. And we are particularly set up for setting your drought stress conditions. We have a feedback irrigation system which allows you to set the drought stress based on the transpiration of the plant, and that allows that each plant has a similar drought stress. You can also set your conditions according to other parameters, but this is one of the most important features. Each unit is independent. That means you can completely randomize your experiment in the greenhouse. The array is set up for many different kinds of plants, from Arabidopsis to large trees, so very flexible which kind of plants can be accommodated. And I have two examples. One is a test for biostimulants under well-watered condition and under drought conditions. And you see the seaweed in green, seaweed provided plants are superior when there is enough water. But under drought, the seaweed provided plants do really significantly fall down in their productivity, while the other product, an amino acid based product, helps with the drought stress, and also those plants recover much more quickly and better. The second experiment done at Wageningen University, testing 2 different quinoa genotypes. And I just want to give you some ideas of data you can get. You can measure over the entire experiment transpiration rate for those 6 different treatments and the water use efficiency. But you can also zoom in. You can actually look at a certain day and a certain hour. Of course, again, we measure constantly, so you get really deep insights in how your plant is doing according to your product or your genotype that you expose to different conditions. In this particular slide, you can see kind of validation of our data in this Wageningen experiment. The team measured manually with a Porometer stomatal conductance and correlated it to the output of our system. And the correlation was 0.95. And if they corrected for the boundary layer resistance, it came actually up to 0.99. In summary, our innovation, we measure directly, we measure precisely, we measure constantly and simultaneously. We measure directly, we have qualitative parameters, so you can really get deep insights in how the plant behaves. And we consider ourselves actually being comparable to an Apple Watch. If you go to a doctor and measure your blood pressure 3 times a year, you do not really get any information. But if you measure constantly, you really have insights on how you are doing, on our case, how the plant is doing. In summary, because you see data right away and you know what your plant is doing, you can have very short testing cycles. We measure many plants simultaneously, we measure directly, we quantify water and nutrient use efficiency. You have a very high resolution, and we can capture, without destroying the plant, the genotype and environment interaction. So you can actually shave off quite some amount of time in your research. So please come see us tomorrow, and I can answer all your questions, 8 a.m. Pacific time. Thank you.
Thank you, Katrine.